All right, welcome to The Explainer. Today, we're jumping straight onto a microscopic battlefield to decode viral pathogenesis. Specifically, we're looking at the exact step-by-step strategies that viruses use to conquer our cells. You know, it's really easy to think of a viral infection as just a simple event, right? You get exposed, and boom, you get sick. But down on a cellular level, what's actually happening is a highly sophisticated, highly strategic invasion. Right this second, inside bodies all over the world, millions of these tiny sieges are playing out. Some viruses, they act like brute force conquerors. Others, they're total stealth infiltrators. So we are going to map out this entire conflict today, going straight to the source material to outline the invasion strategy framework. We'll be breaking this down into 5 distinct phases: the cellular invasion, aggressive viral strategies, the stealth approach, when viruses wake up, and finally, the final outcomes. But before we get into the pure mechanics of it all, I want you to consider a question to frame this whole journey. Why do some viruses, like Ebola or certain respiratory bugs, just rapidly destroy the tissues they infect, while others, like herpes or HIV, can hide inside a human being for literally a lifetime without ever being cleared? Well, the answer lies in the tactical choices the virus makes the moment it encounters a cell. Keep that in mind as we dive into the data. Okay, phase 1, the cellular invasion. So a virus can't just infect any random thing it bumps into. It faces a stark binary choice. It either hits a permissive cell, which means the cell supports the entire viral replication cycle, from breaking in to releasing new virions, or it hits a non-permissive cell. And a non-permissive cell? That is essentially a dead end. It acts like a brick wall that blocks the replication cycle at some stage. Maybe the virus manages to attach, but it just can't get inside. Or actually, maybe it does get inside, but it can't assemble its parts. So why exactly does a cell act like a dead end? Well, usually it's because it's missing specific cellular tools. For instance, the cell might lack the proteolytic enzymes that the virus absolutely needs to chop up its proteins. Or sometimes it's missing cellular transcription enhancers, so the viral genes literally just can't be read. And, other times the fault actually lies with the virus itself. It could be a defective particle that's missing the key genes it needs to replicate. When that happens, the invasion is over before it even really begins. Moving into phase 2, aggressive viral strategies. Now, if a virus does get in, if it strikes gold and finds a permissive cell, it has to choose a path. Cytopathic viruses are the aggressive destroyers. They don't just use the cell, they completely strip it for parts. They actively kill the cells they replicate in. How? By preventing the host from synthesizing its own vital macromolecules, by producing toxic byproducts while they replicate, or by straight up forcing the cell into apoptosis, which is basically programmed cellular suicide. The end result here is total cell necrosis and death. In this destruction, it isn't just random chaos, it is highly calculated. A cytopathic virus might completely inhibit the host's RNA transcription. Or, get this, it might actively degrade the cellular mRNA using its own viral enzymes, literally shredding the host cell's blueprints. Some viruses ruthlessly compete for ribosomes, which are the protein factories of the cell. Others will alter the intracellular ionic environment until it becomes toxic. Sometimes just the sheer buildup of viral structural proteins late in the infection is enough to poison the host. And, like we mentioned, if all else fails, they just flip the switch for apoptosis. It is a complete systemic shutdown of the host survival mechanisms. So the host cell's ultimate fate really comes down to a combination of viral behavior and viral production. If an aggressive cytopathic virus is successfully producing progeny, the cell essentially explodes in necrosis. But if it stalls out and is non-productive, it forces a silent apoptosis. Now, switch gears to non-cytopathic viruses. With these, whether they are producing new viruses or not, the cell actually survives. And this right here sets the stage for persistence, latency, or even full-blown cellular transformation. Which brings us to phase 3, the stealth approach. Let's talk about latent infections. This is where the virus acts like a silent squatter. Unlike the cytopathic destroyers we were just looking at, a virus in latency maintains its genome inside the host cell indefinitely. But, and this is the truly genius part, little to no infectious virus is actually detectable. Why? Because the viral genes aren't being transcribed. There are absolutely no viral proteins floating around for your immune system to recognize. It completely avoids detection by just plain dead. So how exactly does it hide? Well, it chooses one of 2 distinct paths. First, it can become a provirus. This means it takes its viral code, translates it into DNA, and physically knits it right into the host cell's own chromosome. Retroviruses do this, and the host cell literally cannot tell the difference between its own DNA and the viral DNA. The second option is forming an episome. Here, the viral DNA forms a little circular molecule that just floats alongside the host's chromosomes inside the nucleus. It perfectly syncs up with the cell's natural division cycle, which is exactly what papillomaviruses do. The secret to this stealth strategy is actually incredible restraint. Because these non-cytopathic viruses don't interfere with the host's normal protein building, the cell survives. It just goes about its daily business. And every single time that host cell naturally divides to create a new cell, it unwittingly copies the hidden viral DNA right along with its own. The virus propagates persistently, spreading its blueprint throughout the tissue, all without firing a single shot or raising a single alarm. Next up, phase 4, when viruses wake up. The herpes virus is an absolute master of latency, and tracking its physical journey through the body is wild. This is actually the exact mechanism behind why people get recurrent cold sores. First, the virus causes a standard destructive infection in your epithelial cells, say right on your lip. But then, the newly produced viruses enter your sensory nerve fibers. They actively transport themselves up the axoplasm, retreating deep into your nervous system. They eventually settle into the neuronal pericardia, which is the main body of the neuron. Because these particular neurons don't divide, it is the ultimate safe house. Latency develops right there. The virus just sleeps, completely hidden from the immune system until reactivation. Then, it suddenly wakes up, travels all the way back down the nerve, and starts producing infectious progeny all over again. But, this sleeping enemy doesn't just wake up randomly. Periodic reactivation is triggered by very specific, stressful environmental conditions for the host. Things like physical trauma, a period of immunosuppression where your defenses are down, natural hormonal changes, or an intercurrent disease, which just means you get sick with something else. Whenever the host system is stressed out, the virus senses the opportunity, flips the switch from latency back to a productive infection, and launches a brand new attack. And finally, we arrive at phase 5, the final outcomes. When we zoom all the way out, we can see the complete spectrum of these virus cell interactions. At the extreme end, you have cytotoxic infections that cause total cell death and massive morphologic changes. Then you have persistent productive infections, where viruses, like rabies for example, allow the cell to live while just continuously shedding virus. Below that, we see persistent non-productive infections, like papillomavirus in the skin, just waiting silently in latency. And finally, transformation, where oncogenic viruses literally alter the cell's morphology so much that they can go on to produce tumors. It just perfectly highlights the incredible and sometimes terrifying versatility of viral pathogens. I want to leave you with this final provocative thought pulled directly from the text. Integration of provirus may alter the host genotype and the expression of host genes without destroying the cell. Really think about what that means. The ultimate victory for a stealth virus isn't blowing up your cells in some blaze of glory. No, the ultimate victory is quietly slipping into your nucleus, permanently rewriting your very DNA, and ensuring its own eternal survival as a permanent part of you. It's a fascinating evolutionary strategy, for sure. Thanks so much for joining me on this explainer, and keep exploring the incredible mechanics of the microscopic world.